everyone, today we will discuss about nucleic acids, that is DNA and RNA. These are called nucleic acids because these are present in the nucleus of the cell. So this is the cell, this is the nucleus. So these two molecules, these are present in the nucleus. So the DNA, it stays here in the nucleus. It cannot come out into the cytoplasm. RNA, it is made in the nucleus, but it can come into the cytoplasm to perform its functions. Both of the molecules, these are biomolecules. These are present in all organisms. Both of these molecules, they carry informations. First of all, we'll discuss about DNA. DNA stands for deoxyribonucleic acid. This DNA molecule, it carries genetic informations. So what are genetic informations? These are actually all the instructions that an organism needs to grow and to develop from a fertilized egg to a fully grown adult. So this is a fertilized egg, or we call it zygote. So this is the nucleus where it has the DNA. So this DNA is actually controlling how an organism will look like. DNA was first observed in 1800s when many scientists, they doubted that this molecule could carry genetic informations because the DNA, it has relatively simple chemical composition as compared to the proteins, which are much more chemically varied. So in 1953, the experiments showed that this DNA molecule, it carried genetic code. This was the same year when Watson and Crick, they proposed their model of structure of DNA as double helix structure of DNA. So if we see the structure of DNA is actually a polymer of nucleotides. So if this is a single strand of DNA, so the nucleotides, they just appear like beads on a string. So this one bead is a nucleotide. So these nucleotides, they combine together and they make long chain or long strand of DNA. We call it as polynucleotide. So this nucleotide is actually a fundamental unit in the structure of DNA. If we see the structure of nucleotide, it has three components. So now we are going to discuss the structure of one nucleotide. It has three components. So this one is pentose sugar, 5-carbon sugar. The second one is the phosphate group. And the third one is nitrogen containing organic base. Organic base means it has carbon in its structure. So the pentose sugar in the structure of DNA, we call it as deoxyribose because it lacks one of the oxygen as compared to the ribose in RNA. The sugar and the phosphate group in the structure of DNA is the same in all the nucleotides. The only thing that is different, that has different types, is the organic base, nitrogen containing organic base or nitrogenous base. There are four types of the nitrogenous bases. that is adenine, thymine, cytosine, and guanine. So if we see how these nucleotides they join together, so this is one nucleotide and this is just showing the part of single polynucleotide strand. So this is the second nucleotide. So these are the two nucleotides that combine together by condensation reaction between the phosphate group of the one nucleotide and the sugar of another nucleotide. So this is a condensation reaction and this results in the formation of phosphodiester bond because one phosphate has two ester bonds. So these are the ester bonds. So this as a whole we call it as phosphodiester bond. And this structure, that is phosphate sugar in the structure of DNA we call it as phosphate sugar backbone or sugar phosphate backbone. So if we see the structure of DNA, so DNA is actually made up of two polynucleotide strands that are held together by hydrogen bonds between their nitrogenous bases. So if we see this strand, these two strands are held together by the hydrogen bonds between the nitrogenous bases. So if we see this structure, just a model of DNA, these black strings, these are showing actually the sugar phosphate backbone. And if you look closer, that it has a sugar group that is on one side it is attached with the or the nitrogenous bases, these are attached with the sugar carbon number 1, and these are actually the nitrogenous bases which are showing that these are connected by hydrogen bonds. So each base can make a bond with only a particular base. So adenine can make a bond with T, thymine, and cytosine can only make a bond with guanine. So adenine, it cannot make a bond with guanine, or thymine cannot attach with the cytosine. So this is called complementary base pairing or specific base pairing. So there is two bonds between adenine and thymine, and there are three bonds between guanine and cytosine. So there is always an equal amount of adenine and thymine in the structure of DNA, and there would always be an equal amount of guanine and cytosine. And these two strands, these held anti-parallel to each other. If you look closely, one strand is running in this direction and the other strand, the second one, it is running in this direction. So these are called anti-parallel. If we see the structure, this is carbon number 1, 2, 3, 4, 5. So this is the 5 and here if you see, this is carbon number 1 starts from here, 2, 3, 4 and further 5. So this is the 3 and. So here again 1, 2, 3. So this is 5 to 3. This single strand, it runs from 5 to 3, and this is, if you see here, 1, 2, 3, 4, and 5. So this is from 3 to 5. So we can do it with the prime. So 5 to 3 and 3 prime to 5 prime. So that's why we call that these strands, they are anti-parallel. They run in opposite direction. Now we'll discuss about RNA. RNA stands for ribonucleic acid. RNA is quite similar in structure with DNA. One of its main function is just to transfer the informations from the DNA to the ribosomes. So what RNA does, it just take the message. For example, this is the cell and this is the nucleus where DNA is present. So DNA is transferring its information to the RNA, and RNA takes the message from DNA and it comes in the cytoplasm. Where it comes to the ribosomes. Ribosome, actually, they are protein factories. So once the RNA, it comes here with the ribosomes, so ribosome, it reads the message from the RNA, and ribosome, they make the polypeptides accordingly as the message is delivered by the DNA. So actually RNA, it performs this function. It helps to produce these proteins for the cell or for a whole organism. So RNA, it is different from DNA in certain ways. So the it RNA is actually a single strand of polynucleotide, and DNA, it has two strands, or we can say the two polynucleotides. And in the structure of RNA, the nucleotide, it has the same component as the nucleotide of DNA. The difference is, the just we discussed before, the ribose, it here is deoxyribose, but in the sugar is ribose. And another difference is, if we talk about nitrogenous bases, so RNA doesn't have thymine. So instead of thymine, RNA has uracil. So here is thymine in the structure of nucleotide. Another difference is that RNA strand is much shorter as compared to the DNA strand. They are much more longer as compared to the RNA.